Assalamu alaikum. I am Muhammad Kamran Khan and today I am going to present an abstract on effect of selected pyrethroids on feeding behavior of spider. The present research work was conducted under the supervision of Dr. Saida Naseem and Dr. Muhammad Arshad and with the collaboration of Dr. Hafiz Muhammad Tahir. Insects are the diverse group that make 66% of all animal species. Some of the herbivore insects destroy 18% of world agricultural products. When insect population increases beyond limit, they become major pests in ecosystem. Different types of pesticides were used over time to control these pests. Organochlorines are the chlorinated compounds utilized as effective pesticides. It was believed that 40% of all pesticides are made of organochlorine compounds. The most generally used pesticides in Asia that are organochlorine in nature as DDT, aldrin and dieldrin. Organophosphates, carbamates and pyrethroids are also used as insecticides. Pyrethroids are the class of insecticides that are currently used for crop protection worldwide. They are synthetic compounds derived of natural compound pyrethrin. Pyrethroids are poisonous to insects, but they are harmful to mammals, but have agricultural potential also. It is an alarming situation that use of pyrethroids in agricultural fields cause sublethal and lethal effects on natural predators. The first evident change in spiders by contact of sublethal dose showed alteration in movement. Then it reduces the spider's ability of pest control. In the background, in 2012, Packer demonstrated that among all natural predators, spiders are mostly affected by pesticide exposure. In laboratory, more than 40 species of spiders and 130 pesticides have been tested. Results showed that the degree of population reduction subsequent to pesticides application. In 2019, the side effect of pesticides such as bifenthrin, lambda-cyhalothrin and glyphosate on predation mortality and avoidance behavior in spider Neosponathiaceae were studied. About 70% lethal responses in this spider were observed. In India, experiments was performed to observe harmful effect of bifenthrin and malathion on predation of spider. Bifenthrin pesticides residue exposed to spider, they observed that prey capture decreased over time. In the methodologies, first of all, sampling was done. In sampling, spiders were collected from different localities of Jallow Park, Chagamanga, Punjab University and Botanical Garden of Lahore by jaring, handpicking and tree jerking method. The collected spiders were active, same size and without any damage. Drosophila culture was prepared by 2 mashed bananas, 1 mashed potato, 2 tablespoons vinegar and half tablespoon yeast were mixed. Plastic transparent boxes with 6.8 centimeter length and width 5 centimeter were used during experiment. The strip size of filter paper was 15 centimeter in length and 5 centimeter in width were used during experiment. The round piece of filter paper was 5 centimeter to cover bottom of boxes. Preparation of field dose. For bifenthrin, the field dose is 200 microliter per 500 ml and for lambda-cyhalothrin, the field dose recommended was 800 microliter per 400 ml was applied. In the experiment, to examine the effect of selected pyrethroids on the feeding behavior of spiders, 40 adult spiders were selected and divided into 4 groups, each containing 10 spiders. One was control group, second experimental group and other 2 replicates group. The replicate groups were tested the same condition as the experimental group. To standardize the hunger level of spider, they were first offered Drosophila melanogaster flies up to the satiation for about 5 days and then starved for 2 days before using them in the experiment. In the experiment, treated groups were exposed to insecticide treated filter paper for about 1 hour and then placed in the separate clean glass jar. Spiders of control groups were exposed with filter paper dipped in water. Each individual spider was provided with 5 live fruit flies. The predation level of spiders was recorded after 2, 4, 8, 12, 16, 20, 24, and 48 hours of the treatment. Data for the number of prey consumed describes the predation rate. In the results, a table was drawn in the comparison of feeding between bifenthrin treated and control groups of spiders at different interval of time after treatment. In the first column, we have taken time as 4, 8, 12, 16, 20, and 24 hours. In the second group, we have taken control and bifenthrin, and in the third, we have taken prey consumed, their mean and standard error. In the next column, we have taken the value of F test, then significance value, and then degree of freedom, and then the values for the T test. In the second table, we have taken comparison of feeding between lambda cyhaloethrin treated and control groups of spider at different interval of time after treatment. In the first column, we have time 12 or 16, 20, and 24 hours. And in the second column, we have taken groups as control groups and lambda cyhaloethrin, prey consumed, their means and standard error, then the value for F test, significance value, degree of freedom, and its T test values. In the discussion, Brown et al. In 2014 studied the effect of selected pyrethroids on feeding behavior of spiders. Spiders were exposed to field dose of bifenthrin and lambda cyhaloethrin. The results of bifenthrin treatment revealed that the predation of spiders was not affected up to 16 hours of exposure, but with the passage of time, predatory activity of treated groups was reduced. However, no mortality case was observed. Tahir et al. In 2019 observed the effect of lambda cyhaloethrin and bifenthrin on the mortality, avoidance, activity, foraging behavior in Neoscona-thecae. When Neoscona-thecae were offered insecticide-treated prey, their predation rate reduced with time. Results revealed that lambda cyhaloethrin is more harmful to spiders as compared to bifenthrin. In the conclusion, results showed that these pyrethroids were not lethal to spiders. However, some of the temporary effects observed that reduced the spiders' predatory activity, then spiders feed normally after some time. It is the positive aspect because natural predators are safe from harmful effects of pesticides at field dose. Here might be possible that this spider develops some resistance against these pyrethroids. In the recommendations, it is suggested to conduct further research to investigate the effect of different concentration of pyrethroids on activities of predatory spiders and pests at a time. These are the references. And thank you. That is the end of my presentation. Your questions are welcomed, if any.